able to live because of our organs and how they function. For our organs to function, our cells must communicate. The way our cells communicate is called cell signaling. There are 3 steps in cell signaling. Reception is when a signal molecule binds to a receptor protein, causing it to change shape. Transduction moves signal inside of the cell and converts it to a form the cell can actually respond to. After these 2 processes, there is a cellular response that varies with each molecule. Insulin is a hormone produced by the pancreas that signals cells in a specific way in order to stimulate them to take in, use, and store glucose. Glucose is stored in the liver as glycogen. Insulin is released when any of several stimuli are detected. These stimuli include ingested protein and glucose produced by digested food. GLUT4, otherwise known as the glucose transporter, is a protein that accelerates movement of glucose into the cell. The receptor for insulin is embedded in the cell membrane of a muscle, fat, or brain cell. The insulin receptor is composed of 2 alpha subunits and 2 beta subunits linked by bonds. After eating a meal, the food is then broken down in your body and digested. As a result, glucose is released into your bloodstream. As the concentration of glucose rises, the pancreas will release insulin into the bloodstream, which in turn will regulate the concentration of glucose in the blood. It then binds to target tissues such as muscle or fat cells. The first step following insulin binding is when the receptor tyrosine kinase is stimulated. Binding of insulin to the alpha subunits causes the beta subunits to phosphorylate themselves. This process is called autophosphorylation. This event sets off a cascade of molecular events which activate the catalytic activity of the receptor by adding a phosphate group to a nearby protein, which passes the signal from the receptor to the next step in the pathway. Kinases are proteins that add phosphate groups to other proteins. In this specific process, the signal responds to the level of blood glucose and is transmitted from outside the cell to inside of the cell. In the insulin signaling pathway, activation of kinase is one of the earliest steps and plays a major role in insulin-regulated responses. Other molecules will also be phosphorylated. Due to this, other proteins will be phosphorylated as well. This transports the first signal that was sent from outside of the membrane. The next process is the final step in the signal pathway. GLUT4 proteins are embedded in the cell, which allows large amounts of glucose to move into the cell through facilitated diffusion. Through this process, energy is made for the body. However, there are consequences when the signal pathway is disrupted. A collapse in glucose homeostasis can result in diabetes, which is characterized by high blood glucose levels called hyperglycemia. If the disease goes untreated, it will lead to glucose release in the urine called glycosuria. Type 1 diabetes is when the beta cells can no longer produce insulin. In type 2 diabetes, insulin is produced by the beta cells and also binds with the receptor. However, glucose is not able to pass through because the channels won't open.